All right, let's jump right into this explainer and demystify something absolutely incredible. How a static, lifeless chemical code transforms into the actual, physical, moving parts that build our bodies. You know, we hear about DNA all the time, but today we're going to zero in on exactly how it controls cell protein. It's this wild, fascinating journey from a locked-away blueprint to the very structures of life itself.
Seriously, think about it for a second. How does a microscopic string of chemicals actually build a human? Well, to wrap our heads around this, I want you to imagine your body cells as these massive, bustling manufacturing factories. These factories are working 24/7 trying to build incredibly complex products, but there's a catch. The master blueprint for those products is locked away inside a highly secure vault. So how on earth do the instructions get from that secure vault down to the factory floor to actually build the machinery of life? Well, that's exactly what we're going to unpack today.
Here is a quick roadmap for our factory tour. We'll kick things off with Section 1, the blueprint of life. Then in Section 2, we're going to look at reading the genetic code. Section 3 will be all about building the final protein, and finally, Section 4 will explore what happens when the code changes. Let's get right into it.
Starting off with Section 1, the blueprint of life, unlocking the master code.
To make sense of this whole biological manufacturing sequence, we really have to start at the top with the genome. Think of the genome as the ultimate master library. It's the entirety of an organism's DNA. And within that massive library, you have genes. A gene is just a short section of that DNA molecule. You can basically think of a gene as a single recipe. Each gene codes for a very, very specific sequence of amino acids. Now, why do we care so much about amino acids? Because when you string them together, they form proteins. These genes completely control our characteristics because they literally code for everything. They make the structural proteins, like the collagen keeping your skin firm, and the functional ones, like the enzymes and hormones that make your body run.
So, to visualize where this master blueprint is actually kept, picture a cell. Right in the very center, you've got the nucleus, which, sticking with our analogy, acts as the factory manager's office. Inside that office, you'll find these little X-shaped structures called chromosomes. If you were to take a chromosome and unravel it, you'd pull out a tremendously long strand of DNA, tightly coiled up into that famous double helix shape. So the master instructions are kept safely wrapped around proteins and locked tight inside that nucleus vault.
Moving right along to Section 2, reading the genetic code and making the photocopy.
Okay, so this brings us to a massive logistical nightmare for our cell factory. The master blueprint, the DNA, is simply too precious and, frankly, way too large to ever leave the nucleus. It can't go to the factory floor. So the cell comes up with a brilliant workaround. It makes a temporary photocopy to send out instead. This photocopy is called RNA. Notice the differences here. DNA is a big, double-stranded double helix, and its chemical alphabet includes a base called thymine, or T. But our temporary photocopy RNA, it's much smaller. It's just a single-stranded molecule, and crucially, it completely drops the base thymine. Instead, RNA swaps the letter T for a different nitrogenous base called uracil, or U.
We call the process of making this photocopy transcription, and honestly, the way it unfolds is a biological marvel. First, a section of the DNA molecule literally unwinds, breaking its bonds to expose the template strand. Next, free messenger RNA, or mRNA, nucleotides that are just floating nearby, swoop in and bind to their complementary partners on that exposed DNA. They lock together to form a single continuous strand. And just like that, boom, the brand new mRNA photocopy sneaks out of the nucleus through a tiny pore.
It is just incredible to see this microscopic machinery in action. An enzyme unzips the DNA strands, and you can see the free RNA nucleotides matching up perfectly with the exposed template. The A's, U's, C's, and G's are pairing up one by one to assemble that brand new single strand of mRNA. And then, once it's built, you see the finished mRNA escaping right through the nuclear pore and out into the cytoplasm. It's this flawless, high-speed photocopying process that is happening constantly inside of you right now.
All right, on to Section 3, building the final protein. Time to hit the assembly line.
Let's move out into the cytoplasm for the actual physical manufacturing phase. We call this translation. Once our mRNA photocopy gets out here, it attaches to a ribosome. Think of the ribosome as our main assembly machine. Now, to build something, we need raw materials, right? Here, our raw materials are amino acids, and they get delivered by transfer RNA, or tRNA, acting as our busy factory delivery workers. Each tRNA molecule carries a specific amino acid at one end, and at the other end, it has a triplet of unpaired bases called an anticodon. As the ribosome chugs along the mRNA, the anticodon pairs with a complementary triplet on the mRNA called a codon. When two tRNA molecules line up side by side, a peptide bond forms between their amino acids. This whole process just keeps going until the ribosome hits a designated stop codon.
Now, the real magic happens right at that pairing stage. The tRNA anticodon acts exactly like a key, perfectly locking into the three-letter mRNA codon. That perfect fit ensures the exact right amino acid, whether that's methionine or tyrosine or whatever the recipe calls for, is placed in the exact right order. As the ribosome moves down the mRNA strand, it literally strings these amino acids together into a growing physical chain, all linked up by peptide bonds.
So once that stop codon is reached, the factory line stops and the chain is released. But wait, it's actually not a working protein just yet. This newly finished amino acid chain now has to fold and twist itself into a highly specific 3D shape to form the final protein molecule. This folded chain is what literally becomes the physical stuff of your biology. It's the collagen giving your skin its structure, the enzymes digesting the lunch you just ate, or the antibodies circulating in your blood right now keeping you healthy. This is the exact moment the chemical DNA code physically becomes you.
Finally, Section 4, when the code changes. What happens when there are typos in the blueprint?
So, what goes down when there's an error in our carefully copied factory instructions? Well, we call these typos genetic mutations. They're basically rare, random changes to the DNA sequence, and there are three main types. Insertions, where a brand new base is randomly jammed in. Deletions, where a base is just completely removed. And substitutions, where one base is swapped out for a different one. Now, a substitution might only change a single amino acid, but insertions and deletions, they are incredibly disruptive. Because remember, the mRNA code is read in groups of 3, so adding or removing just one single base has this massive knock-on effect. It's like skipping a bubble on a Scantron test. It changes every single 3-letter grouping that comes after it for the entire rest of the gene.
Now, a lot of the time mutations don't alter the protein enough to really matter, but sometimes they have severe physical consequences. If the DNA code changes, the sequence of amino acids changes. And if that sequence changes, the way the chain folds into its final 3D shape changes too, which means a vital enzyme might end up with an active site that is completely the wrong shape. If the active site changes shape, its target substrate can't bind to it anymore, and suddenly that crucial enzyme is entirely useless.
Which leaves us with this final, honestly mind-blowing thought. In a genome made of billions of letters, a single inserted or deleted base can completely derail a structural protein or totally break a vital enzyme. Yet second by second, in trillions of cells inside of you, your body's RNA photocopiers and ribosome assembly lines are churning out the proteins that keep you alive, basically without fail. When you really stop and think about it, isn't it absolutely incredible how often the cellular factory gets it perfectly, 100% right?